stages that embryos go through. And we've talked now about fertilization, cleavage, and ejaculation. And today I want to introduce a new element to this, which is the patterning of the body in terms of polarity. Anterior, posterior, dorsal, ventral, left, right. When do embryos start to establish this? And how does that tie in with some of the other topics we've talked about so far? And mostly today we'll be talking about frogs or newts. So both of these are amphibians and are vertebrates. And I wanted to start with a movie to talk about the cytoplasm here of the frog. If I can get my cursor to behave. So at the beginning you see up here in the animal pole of the frog embryo, there is some darkly pigmented cytoplasm, and then down in the vegetal pole where it was more yolky, the cytoplasm was a different color, it was more gray or clear. And what you saw in that video, I'll show it again, but is after the sperm had come along and fertilized, on one side of that embryo, now there's a literal shift or movement of the cytoplasm just on the very outside layer or cortex. It's still all one cell. So just the outside layer of that gray pigmented animal cytoplasm moves about 30 degrees away from the site of sperm entry. So let's have another look at what that looks like. Bigger. Okay, see how it's pulling back? So the sperm would have fertilized here to the left-hand side of the screen. Okay. Okay. So, I'm used to this curve thing. Okay, so initially, before fertilization, we have an egg that is essentially radially symmetrical. So we could draw a line from north to south pole or animal to vegetal pole. And around this circumference, everything is basically symmetrical. There's not really any left or right side, dorsal, ventral, or anything. But once the sperm comes along, it is going to fertilize somewhere on that egg. And in doing so, it breaks the symmetry and gives that egg, or that now fertilized egg, some information that it will use as it begins to build the body. I need a little more room to walk around. So here you see the cortical cytoplasm is going to shift 30 degrees towards this side where the sperm has entered and fused with the egg plasma membrane. So this sets up the first body axis, which happens to be the dorsal ventral axis in the frog, and the side opposite, where that sperm happened to fuse, and it could be anywhere, the side opposite will become the dorsal side of the future embryo. So you can't really tell that right now because we're still in the one-cell stage. But cleavage is going to happen, ejaculation is going to happen. You'll soon become a blastula with many different cells. And this side opposite where that sperm had originally fertilized, that will begin to take on the character of the dorsal part of the embryo. Okay? So this begins with the shifting of cytoplasm. Another thing that we talked about last time, you guys did an in-class exercise on, is the idea that the mother is putting RNAs and proteins into this egg. And they are not necessarily evenly distributed throughout the egg. In fact, they're not evenly distributed. So one of these, just an example, is a protein called disheveled, DSH, represented by little blue dots. Initially, that's found more down in the vegetal pole here. But after this cortical cytoplasm moves, those disheveled molecules are going to move along with it and get brought up here to this side that will become the dorsal side. So that's just one example of a component that now becomes sequestered or localized in a very particular region of this one-cell embryo. And that happens as a result of cortical rotation of the cytoplasm. Okay, so this basically gets at the same sort of idea here. So we have material components are being moved around within that one-celled embryo. Later, when we get to the blastula stage embryo, multicellular with a blastocele cavity, there will be developed two signaling centers that are needed for further patterning of this egg and needed for building the anterior and posterior body axes. And these two signaling centers are called the Newcoop Center, which no one could ever spell. It's named after the Dutch investigator who originally did the experiments I'll tell you about today that discovered this center. And then the second place is called the dorsal blastophore lip, and that's the site that last time we talked about gastrulation initiates here. So this is the first site where cells are on the outside of the embryo and they're beginning to move inward into the blastocele cavity. So that site in itself has a number of very particular specialized molecules that are providing information to the embryo and are going to start helping say this will be the anterior side, this will be the posterior side. So in the blastula stage embryo with the blastocele cavity, the location of these two signals is here on the side opposite of sperm entry. So by convention, we'll draw always this left-hand side as being the side where the sperm happened to enter. So opposite that are these two signaling centers, the Newcoop center and the organizer, or the dorsal lip of the blastophore. This is also called the Spemann organizer. So Spemann organizer, after the discoverer, Hans Spemann, and the dorsal lip of the blastophore, the same thing. So these two are located here with, basically they're right next to each other on this dorsal side. So eventually, this is not, frogs and newts they're not shaped like spheres. They're shaped more like elongated rod-type structures as embryos. And I've just drawn here, dorsal will be the back side, ventral the belly side, anterior with the head, and posterior with the tail. Okay, so after the sperm has entered or fused with the cytoplasm of the egg, we had our cortical rotation, and we're getting ready for the first cleavage. So this slide is to demonstrate the orientation that cleavage takes. And the dotted line there is showing you where that first cleavage will occur. And it's basically going to be along the lines of where the sperm had fused, and it'll divide the embryo in two. And this will provide two cells that actually are becoming the left and right side. So then when we get another cleavage, it'll be offset 90 degrees from that. So this will divide the embryo into 4 cells. So we'll have the left and right side, and then the following cleavage will divide it into cells that are on the ventral versus dorsal side. Does that make sense? I think I have another slide here. Yes. So this shows you, with relationship to the cortical movement here, the 30-degree rotation, that first cleavage will actually bisect where that cortical rotation had happened. So that little white or light gray area shows you the cytoplasm that was underneath the pigmented cytoplasm. This is cytoplasm that's revealed when that cortical shift happens. And the very first cleavage is going to bisect that. So each daughter cell gets sort of half of the components that are in the cytoplasm of the egg if we divide it this way. So one question you may ask is, are these maternal components present in the egg at this point? Are they actually critical for establishing the body axis? And a lot of these experiments that I'm going to tell you about today were actually done some time ago by the classical developmental biologists. And they really didn't know a lot about what they were doing, but the experiments were pretty informative. Just as simple experiments to find out questions about body patterning and when and how it happens. So the first very simple experiment is just to say, okay, let's separate these two cells and then keep them in a dish in an incubator, say under conditions where they could continue to divide survive as cells and to divide to give daughter cells, and see what sort of embryo you can get out of that daughter cell. So if you separate the two cells and then allow them to develop on their own, they do actually survive, continue to make daughter cells, and each of them will make an embryo that has pretty much normal body patterns. So that would imply that each of the daughter cells of this two-cell stage here, each of them has enough information to go ahead and decide a dorsal and a ventral and an anterior and a posterior situation as time goes by. But if you do the experiment a different way, if you flip this embryo around 90 degrees and you take a little piece of let's say a hair, because you're back in 1900 and you don't have fancy sutures or whatever. So Newcoop actually took a hair off of his baby daughter's head to do this experiment, just because it was a very fine, gentle ligature. And he wrapped this around the embryo and forced it to divide in a 90-degree offset situation compared to what it normally would. So that one cell got the entire contents of that side opposite the sperm entry, and the other cell did not get any. And in this case, when he separates the two cells, something very different happens. The cell that got the cytoplasm opposite the side of sperm entry, that guy became a normal-looking embryo with a dorsal and a ventral pattern. And then the other daughter cell became this belly piece, which is basically a blob of tissue that really resembles cell fates that are ventral, that is on the belly side of the embryo. So it lacked something essential. So that meant that in this side opposite of the sperm entry, there must be some kind of critical factors or components that the embryo needs to make the dorsal side. Okay. So that already flags that area as being an area that's special, an area of interest for the embryo to go ahead and start body patterning. Okay. So then Newcoop here, he went on to do another set of Thames experiments to again try to test where are these signals in the embryo and what are they doing. So this set of experiments was done on blastula stage embryos. So you can see here they have the blastocele cavity. And on our slide they've been color-coded because we are telling you what the cell fates are for these daughter cells. But at the time he did not know this information. So what he did is to slice off the very tip of this embryo, and he called that the animal cap, because it's at the animal side of the embryo, and it's sort of just a little cap of cells there. And then he sliced off the bottom half of the embryo, and he called that the ventral half, the ventral cap. And so he's eliminating these cells that are right at the border between these two portions of the embryo, the cells colored in red, which we now know are mesoderm. And so he took this animal cap and ventral cap and he put them together in a little sandwich and looked to see what happened. Now normally, if these animal cap cells were left on their own, they became ectoderm. And he could tell this by looking at the structures that developed. But if the animal cap cells were placed in contact with the vegetal half, they changed their cell fate and became mesoderm instead, all of them. So all those cells underwent a total shift in life planning, and instead of becoming ectoderm, now they became mesoderm. So this implied that there must be some kind of signal coming from the vegetal cells that if the animal cap cells saw that signal, they would become mesoderm. But if they were separate from that signal, then they would stay as ectoderm. So this is the model. And that brings us to a really important concept in developmental biology, which is the concept of induction. And what this involves is one group of cells sending out a signal to a second group of cells, and then based on that signal, that second group of cells changes their cell fate. And so on this slide, we have some features here about the inducer cells. So for example, the vegetal cells in this experiment, they are the ones that sends out the signal to the inducee, if you will, the mesoderm cells up at the top, the red cells. They themselves, the vaginal cells, are not changed. So even though they send out the signal, their own cell fate doesn't change at all. They just stay as vaginal cells or endoderm cells. And yes, I guess I'm repeating myself here, they're not really changed by the induction process at all. So it is a kind of a one-directional signal from one group of cells to a second group of cells, where the second group of cells gets changed. And we find many, many examples of this in developmental biology, and it's one of the reasons that gastrulation was important, because remember how cells got brought into new positions? So now that they're in the new position, they're in a close localization to tissues that might be sending out an inductive signal. So they're able to receive that signal and therefore change appropriately. Okay, this also, if you think about it, means that the blastoceal cavity is important, because the blastoceal cavity is maintaining a separation for some of the cells up there at the tippy top of the embryo of the animal calf, so that those cells can be free from an inductive signal telling border cells to become mesoderm. So this signal coming from the vaginal cells here, any cells around the periphery, those can receive the signal and be induced towards a mesoderm cell fate. But the very animal cap cells separated by the blastoceal cavity, those don't see the signal, and therefore a portion of those remain to become ectoderm, which is what we want. In addition, there's another layer to this. And you'll often find there's lots of layers in development. If we look at these animal calf experiments here, and we can classify all the fates that develop from these animal cells as mesodermal cells, but they're not exactly the same kind of mesoderm. There's some pattern within that as well. So over here on the side opposite spiramentary, we see mesoderm that's more dorsal in character. And for example, that's going to be somites and notochord. And these are two structures that are kind of characteristic of mesoderm in the dorsal portion of the embryo. And we'll find out later that somites become muscles and connective tissue in the back. And notochord is a temporary structure that runs along the very back of the embryo. If we look more in the middle, then that mesoderm was definitely still mesoderm, but was more intermediate. And these had fates such as organs that you might find kind of in the middle of the body, muscle that you find in the middle of the body. And then over here, where way back when the sperm had fertilized, on that side the mesoderm was more ventral in character. And you would find things like blood and mesenchyme tissues that you normally see on the ventral side. So if we're going to explain this by one or more inductive signals, we have to think, what can explain mesoderm development and what can explain this regionality or this polarity across one side to the next? And so we're probably going to need at least two signals. One signal that would say become mesoderm, and then one or more signals that would be found across the embryo, say be dorsal, be medium, be ventral in character. Right? Just thinking out loud how this might work. Okay? So to explore that idea, you can take this a step further, and he began to use, instead of entire vaginal halves of embryos, he would separate this out to only use, say, one daughter cell at a time, or one or maybe two. And so you could do this if you took embryos at a fairly early stage of cleavage where the cells were already cells but still pretty big, so you're kind of taking one cell that represents a decent chunk of territory from the embryo vaginal half, and see what that could do for these animal caps. And so here you see 4 different cells. They each have a name, D1, 2, 3, 4. And he would place one or more of these daughter blastomeres in contact with an animal captain, you see what you get. You can score them, is it dorsal, intermediate, or ventral in character. And the D1, that's the blastomere that is opposite the side where the sperm originally was fertilized. And you can see that gives 77% of the time it gave very dorsal characteristics. 23% of the time it gave kind of intermediate mesoderm. And really almost never gave a ventral characteristic. And then progressively, as you move across the embryo here, you get more and more ventral in character and less dorsal character. And so this kind of confirmed what he was already thinking, that there may be some special signals over there on that side of the embryo that's opposite where the sperm had fused, and that those signals are needed to indicate the degree of dorsalness as this embryo developed. So I think it's kind of clever the way that you can figure all this out just by little simple experiments like this. Okay, so now we have basically what I've called show-it type of scientific information. We have a correlation. You can see that there may be some kind of special properties over here on this side opposite sperm entry, and that those properties correlate with the development of a dorsal type of cell fate. And so, since this seemed to be a specialized region, June24Part1 gave it a name, and I don't know if he named it after himself or if that just became the name, but this is called the June24Part Center, and it refers to a group of cells down here, opposite side of sperm entry, that are involved in creating this dorsal side of the embryo and may contain signals that are needed for the dorsal side. So the D1 blastomere, that would be the progenitor blastomere that originally, or that as time goes on, are going to give rise to multiple cells in this area. Is this going too fast? Are we okay? Okay. You can slow me down. You can interrupt with questions, whatever. Okay, so now let's take it to move it and block it types of experiments. So we're taking the next step in scientific inquiry here by interfering with the system and then testing if something necessary, is something sufficient, by manipulating what goes on. So this is going to be a move-it experiment, and it's going to involve a host embryo and a donor embryo and a D1 daughter blastomere, because we know that guy is special and may have these particular properties to it. So in yellow here, here's your recipient. And what would happen is this D1 blastomere from a donor got transplanted over here in the site where the red cell is. So now this embryo has two D1 daughter cells. It has the original one, or the endogenous one. So endogenous is a word that means what is naturally part of the embryo, its own version of it. And then we have this red one, which is the donated one. And it's in a different position than the endogenous D1 blastomere. It's kind of on more towards the would-be ventral side. And the result of this embryo, if you allow it to go on, form a blastula, form a gastrula, and keep on developing, it becomes this embryo with two heads. And that's kind of exciting for people who are interested in trying to figure out patterning in the embryo and how we get a dorsal side and ventral side. But you can mess this up to the extent you actually get two heads. Something interesting is going on here. So just by moving this one cell, that was sufficient to duplicate the dorsal side. So I know this is not purely dorsal side, it's also kind of a duplication of anterior fates, right? Because that's head. But in the embryo, you establish dorsal ventral, and that is kind of happening a little bit simultaneously with the anterior posterior. So you get that as well. So this definitely confirms there's something special about signals coming from the D1 blastomere, and that if you put those signals in, that's sufficient to generate the dorsal side of the embryo. You put two cells in, you get two dorsal sides and two heads coming out of this. Okay, what about blocking? So usually if we do a move-it experiment, that is more designed to test if something is sufficient, whereas if we do a block-it experiment, that's more designed to test if something is necessary. So think back about there's a distinct meaning between these two words, being sufficient, that thing by itself can accomplish the goal, or being necessary. It's one of possibly many factors, but you have to have this particular one in order to accomplish the goal. Okay, so a block-it experiment, and again, the method doesn't matter. It could have been a mutation, it could have been a chemical, any kind of treatment that we could put upon this poor little embryo to block the effect, then we can see what happens in that scenario. So in this case, we're going to use UV radiation. And what UV radiation will do is prevent that original cortical rotation. So if you shine UV light on these frog or newt embryos, now, even though the embryo gets fertilized, you never see that shift in cytoplasm towards the side of the sperm entry. And as a consequence, you would never see the rearrangement of the maternal factors that might be needed to establish the nucleus center. And if you let that thing go, sure enough, it develops just as a body piece. And essentially what it is an embryo lacking the information for dorsal ventral sidedness. And the default state here is just to develop as a ventralized embryo or a belly piece. So we're really lacking the information to build the dorsal side. Okay, as a control, you always have to have a control. If you go ahead and let that embryo develop in the same dish, but you didn't shine the UV light, then you get a normal embryo. Okay, so if we're blocking that, now we don't get dorsal sidedness, so it also supports this hypothesis that something special about the D1 blastomere following cortical is needed to establish. You have to actively, proactively establish the dorsal side in these embryos. Okay, now I throw a little twist at you. Suppose that we did this experiment. We treat the embryo with UV. Then we do the transplant experiment. What would happen to this embryo here? So we've done the transplant experiment just like before, where we moved it not to the site where the endogenous D1 is, but to to a different place, more towards the presumptive ventral side. What would happen in that? So why don't you take 2 minutes, think about it, talk to your neighbor, and then we'll come back here and see what happens.
So, we're going to transplant part develop whatever the transplant part was, because everything else is going to be like.
It's going to be hard. I don't think it's going to grow.
What are you saying?
I don't think it's going to cut them off.
Yeah, as a whole, so just that transplant section.
Yeah.
Like not. You don't even. No.
Like fix it. I played the soccer game last night. We lost.
Yeah. That's cool.
4v4.
Huh?
4v4.
4v4? Oh. Gotcha.
Okay, who would like to offer a hypothesis about what's going to happen here?
I think it will develop more.
Okay. So would any part of the embryo be different than the wild type, or different than the control?
I don't think so.
Okay. Anyone else have a thought?
I just think that because it's on the other side,
Yeah.
it will be formed in a way that the head will be more ventral, not dorsal.
Okay, so you're wrestling with the idea that you put this on the other side.
I just don't know what would happen.
So what do other people think about this, putting it on the other side? Yeah.
Would it just flip what dorsal and ventral is on the organism?
Yeah. Yeah. So I think the three of you each got elements of this. So if if we do the UV, we're getting rid of the endogenous dorsal ventral information, right? So we're wipe it clean. Now we have what would be a belly piece if left by itself. Then we come along and we add back information to form a dorsal side, but we put it on the other side. So it's over here. So it's just as if the original sperm might have fused on this side, the right-hand side, instead of the left-hand side. So it will develop normally, you're right, but it's going to develop, flipped from what the control would have been. I mean, they're just the same. They're actually the same, but yeah. So it is essentially normal, but because we put this on the other side, it's as if the sperm had fertilized over here instead of had fertilized on the left-hand side. Yeah.
Would there be any other effects on the embryo from using the UV on it? Because it's my understanding that UV can also like mess up your DNA. So would this embryo that now developed with the flipped body plan, but still more or less correct, have any other issues just because it was originally treated with UV?
So that's a good point. And these are not perfect experimental tools. And so what is happening in with the UV, I've got a slide here in a minute too, is disrupting microtubules that are important for moving that cytoplasm. And we're kind of ignoring the fact that it might have actually caused mutations in the DNA or something like that, or that could happen, and that might then cause that embryo to develop abnormally. But that's not particularly informative for what's going on with this experiment, but it's a good explanation.
So it wouldn't have any effect on that, inserting the D1 blastomere then?
Right.
Okay.
Yeah. So, when you do experiments like these, you might do it 100 times, and let's say not everyone is going to be informative, and not every time would it develop normally for sure, but that's due to experimental manipulation problems. But it has the potential to form a perfectly correct embryo. And that embryo will be normal for left-right sidedness. It's just that kind of shows you this fertilized egg had the potential to develop different portions as the left or the right side, depending on where that sperm was fertilized. Any other thoughts or questions on this? Okay.